Hello, everyone. Welcome back to Creation Myths. We're here live tonight for what I anticipate is going to be a really fun and a really content-packed kind of show. And I'm excited because we're going to be talking about things that I sometimes don't talk about that much. There's stuff in here that I get to learn a lot about tonight. So my guest tonight is Andrew Cumming, also known as a rational empiricist. You've probably seen him making the rounds in various evolution and creation discussions on YouTube over the last few years. He was actually one of the first guests on my channel, like way back when I was still doing this in my old office on my old laptop. And so I'm really happy to have Andrew back. Andrew, tell us anything else you want to tell us before we get into things.
Yeah, so thanks for having me on. My name's Andrew. I'm currently a college student hoping to transfer over to major in biology at a major university soon. I also have interests in cosmology, paleontology, geology, generally the general creation evolution topics out there. Yeah, that pretty much sums it up. I hope everyone enjoys the stream tonight and maybe learn a thing or two, and hopefully use some of the arguments we're going to talk about tonight.
Yeah, so tonight's topic is, I think, really interesting. And this was Andrew's idea. We've been talking for a while about getting back together and talking about stuff online. And so Andrew came up with this idea, and I love it. So the topic tonight is that we're going to look at some of the major pieces of evidence for universal common ancestry or universal common descent among cellular organisms that you don't often hear about, right? So you hear a lot about phylogenetics, you hear a lot about looking at the fossil record going back and looking at how, despite what creationists say, we actually do see a lot of transitions between major groups in the fossil record. We also can see gradients genomically as we do these comparisons, and we can see the relatedness and the nested hierarchies among
Or
the major
ERVs,
groups.
for example.
Exactly. So these are the things that I talk about this stuff all the time, but there's tons of other things that don't often get discussed as much. So we're going to touch on just a few of those things tonight. So with that said, let's Andrew, take it away, because you're really in the driver's seat tonight. So which paper should I pull up first that we want to dig into here?
Yeah, so the first paper I want to talk about comes from 1992 by Hillis et al. It's a major study looking at basically, of course, we rely on phylogenetics to talk about organisms in the past, organisms today, their phylogenetic histories. And of course, we want to be reasonably sure, as much as possible, that the methods we're using to construct these phylogenies or infer these phylogenies from genetic sequences or morphological characters are reliable. You want to be able to trust the methods, obviously. So what the researchers here did, there we go, Hillis et al., 1992 did, was they took these often used phylogenetic methods and basically had cultured and repeatedly split some viruses, specifically bacteriophage T7, with a kind of mutagen present. So of course that would cause mutations. And as the viruses kept splitting, basically speciating effectively, those branch points, each lineage was tracked. So you have this known phylogeny, essentially, because we saw the viruses branch out and happen. So we have this known phylogeny that was directly observed. Now, what they did was they took 5 independent phylogenetic methods, some of the ones commonly used, so things like parsimony and maximum likelihood, and compared based on the data at the end of the sequence, the genetic data based on those mutations, seeing if they could track the lineages back if those phylogenetic methods could construct the correct tree topology based on the data they had there. And it turns out that out of, I think it was like roughly 135,000 or so possible phylogenies, all 5 of them came up with the correct tree topology. So that shows that these kinds of phylogenetic methods that we commonly use, they are reliable. They can construct or be used to infer known phylogeny. So we have uh, we're rational. We can trust them going backwards in time to unknown phylogenies, basically.
Exactly. This is experimental validation of kind of the foundation of phylogenetic techniques. And this was done in 1992. So like our sequencing technology was more primitive, our computational abilities were not,
Right.
what they are today. So the data set is smaller, the genomes are smaller, right? We're comparing fewer nucleotide sites. But what this paper shows, and I will say this is of the papers that Andrew brought to talk about, this is the only one that I'm familiar with. My background is experimental viral evolution, so I deeply love this paper. Just the idea of saying we're going to split these viruses, we're going to have a known phylogeny, and then we're going to sequence them, and we're going to do the phylogenetic techniques and see if we actually get what we know to be the correct answer is great, because it validates those techniques. And for people watching, you may be, if you're familiar with creationists when they talk about phylogenetics, what's the phrase on your bingo card for creationist phylogenetics? Lines on paper. Just lines on paper, right? But no, it's not just lines on paper. We actually have experimental validation that when we do these comparisons and we show these relationships, what we're actually uncovering are true evolutionary relationships. It's not just spurious associations.
Right. They're
Right.
not
So this
just
is
statistically or mathematically robust, they're also empirically solid.
exactly. So this is a really important paper to have in your back pocket to be able to, when somebody questions like just phylogenetics as a whole, right? Here you go. No, we've experimentally validated these techniques. So we can actually like
Right.
be confident that they're actually showing what we are saying they're showing.
And I
And
will
that's a
also
really
add,
important thing.
right, and I will also add on the subject of viruses and virology in general, for those who are creationists who think phylogenetic methods are uh, imprecise or unreasonable, every time you get like a flu vaccine next year, or when you're developing vaccines in general, or tracing the history of a particular viral lineage, seeing what was the patient zero, basically going back, like constructing these overall virus phylogenetic relationships, those all rely on these same methods. So if they weren't reliable to some degree, then a lot of medicine and viral genetics and scientists would be out of kind of out of a job there, or they wouldn't be able to give us these accurate uh, medical conclusions, basically.
Yeah, there's a fantastic paper from, I believe it's from 2010, but it's the phylogenetics of the 2009 swine flu pandemic. And it
Um,
has this amazing figure of, it's this big, dense phylogeny with hundreds of lineages on it based on hundreds of sequences. And if you look at the time axis on that phylogeny, it just goes from January to like maybe even just as short as like July or August of 2009. So it's just like the early phylogenetic history of the 2009 pandemic. And that's important because we want to know where that virus has been and where it's going, right? So you want to be able to accurately reconstruct its actual pattern of descent. Well, how do we know that we're actually doing a reasonably accurate job of that? It's because of things like this, where we have experimental validation of those techniques. So we can do it in the lab, show that it works, and now we can apply it to these other situations. And like you said, Andrew, we're talking about viruses here, but these techniques don't just apply to viruses,
right.
and it's using a viral model organism to show the validation and then helping bolster our confidence when we use them in other contexts.
I mean, it's like what Erica Gutsit-Gibbon has frequently said in some of her videos: unless the Mendelian laws of heredity stopped working at some point, and maybe some creationists want to propose that, but at that point, that's kind of appealing to miracles, then there's no good reason. In fact, empirical evidence based on papers like this, that we have good reason to trust these phylogenetic methods going beyond the species level, the family level, etc.
Yeah, that's a great point, that you can take these techniques working backwards and you see the same level of confidence in those results that you see when you're looking at, for example, experimental viral populations. And if we actually had, in reality, separate ancestry where the families or the orders or whatever level, wherever you want to draw the line, right, if these different groups were actually separately created, at some point as you go backwards in time and increase the diversity of your comparisons, it's just going to fall apart. You're not going to get a nice, neat tree like this. You're
Right.
going to get gibberish. But that never happens, right?
Right.
You still
And
make that
just to
nested
kind of
hierarchy.
right, and just kind of to add on to what you said there, in fact, if separate ancestry were true, you in all likelihood wouldn't just get like a polytomy kind of thing where you different tree topologies, different branching histories aren't one's not well supported over the other, which in for some clades you do find that, but for a lot of other clades, of course, you do find strong congruence. Um, but like if separate ancestry as a whole were true, then you'd see conflicting trees all over the place. Like
Exactly.
there wouldn't be this high statistical significance of the amount of congruence that we do see. So that itself is another strong argument for common ancestry
Bingo.
that we can't
Absolutely.
really touch on here. But
Yeah, totally. That's a great point. Was there anything else you wanted to add with this paper? Because we talked about this one actually just being a short little intro into
right,
some of the other ones. So should I pull up paper number 2, which,
yep,
if I
yeah,
recall, was the do you want to do cytochrome C? Is that the next one?
that's the third one.
That's the third one. What's the second one
So
you want to
the
do?
next paper
I'll put this
is
one down.
Benton et al. Or
All
Benton
right.
and Hitchin,
Ah,
1997.
yes, great. Let me pull that up. Okay,
So
we are going to.
the next argument we're going to be discussing is the actual remarkable amount of congruence, because when you talk to creationists, they'll say things like, well, the flood uh, ordered the fossil record basically based on ecological zonation or hydrodynamic sorting, which those don't actually work by themselves, but that's a whole other discussion.
And
Um,
also, trees can't like run uphill.
right, a
Like
whole
all the
thing about
flowering
plants.
plants climbed the mountains, but the stupid ferns stayed down below.
Right. Yeah, taking that as a hypothetical given here for sake of argument, if that were the case, you would probably expect the pattern that we see in the fossil record, and that is the order and or the first appearance and last appearance of specific fossil clades, basically fossil groups. You should expect them to kind of be to to follow those kinds of patterns. Basically, they shouldn't follow a branching tree, the pattern that we would expect from common descent, because you have the processes that govern this supposed global flood and the processes that we know govern common descent and the population mechanisms that we see today. So those are kind of independent of one another. So if you can compare the two and see, okay, do they match up? Does the stratigraphic first appearance and last appearance, the order basically of the fossils, generally speaking, in the fossil record, in successive rock layers, does that match the phylogenetic divergence patterns that we see for life in general? Like you would, like based on the standard phylogeny, for example, you would expect to see the common ancestor or relatives of the common ancestor of birds, mammals, and reptiles before the first appearance of birds or mammals or reptiles, for example, because certain clades, in a sense, have to come before others, or the branch points have to come before others. I hope that makes sense to
Yeah,
everyone.
this is, I mean, we're talking about nested
Yeah,
hierarchies and common ancestry. So you have
right,
to have the common ancestor before you have the subgroups of any of the descendant clades,
right,
right? So you have to
exactly,
have your mammal reptile common ancestor before your bird lizard common ancestor, for
right,
example, right? And we can phylogenetically, based on existing genome sequences and comparisons
right,
using the
living organism,
phylogenetics we just talked about, you can phylogenetically calculate when those things would be. We also have a robust fossil record of those things. And we
right,
should, right? Our hypothesis, our expectation should be that those two independent data sets, as the authors say right here, should match up with each other. That's what we would expect if we basically have the right idea of what happened.
Right, and this could have been a falsified prediction, basically, because on the one hand, the fossil record is still notoriously incomplete. There are still arguments over the quality of certain parts of the fossil record, and also, as we mentioned before, some parts of the standard conventional tree of life are still a bit unresolved. So you kind of have to take both of those together when going into this analysis, when kind of weighing one data set against another. But yeah, in this study by Benton and Hitchin, 1997, basically they compared the if you can go to the discussion portion, I believe.
Oh yeah, sure. Let's down to the, let's see, that's the results. Here we go, discussion. Here you go.
Okay, and basically, I believe it was towards the end of the discussion, if you don't mind zooming in a little bit.
Keep going.
As well,
Oh,
so
there you go. That's down here. Here you go. So this is the end of the discussion down here. That's the last page.
Okay, yeah. So basically, is there any way to zoom into that specific portion?
Like where? Like this part?
Yeah, that part specifically.
Let's zoom into that part. That's probably too big. There you go.
There we go. Okay, that's all right. So yeah, basically what they did, what these two researchers did was they compared like we said, the stratigraphic order of fossils against 384 published cladograms or phylogenetic trees for echinoderms. So that's like sea stars, sea urchins, sea cucumbers, various kinds of fishes, and tetrapods, using 3 different metrics. You don't really need to know what those specific metrics are. They're basically just ways of analyzing the amount of correlation between, like we said, is there a positive, is there a negative correlation between the stratigraphic order of fossils and the divergence pattern of modern organisms, or is there a negative correlation, that sort of thing. So measuring the amount of agreement, basically, how well do they match up. And what they found was overall, these the most of the cladograms were statistically significant, or the amount of congruence between the fossil order and the modern phylogenies was very statistically significant. Looking at my notes here. So according to a 2003 article by the same author, Michael Benton, titled "The Quality of the Fossil Record," basically there were some earlier studies than this particular one. This was in 1997. So for example, there was a study by paleontologists Norell and Novacek, 1992, that looked at mammals specifically. And there have been studies since then, including this one, that have tried to assess, again, what is the quality of the fossil record, does stratigraphy and phylogeny match up? And what Benton wrote here in 2003 was basically that these later studies since 1992, or Norell and Novacek 1992, basically 36 to 50% of the trees in this particular study were congruent with fossils based on what's called the Spearman-Rank correlation specifically. So that's one of those metrics, SRC, how you can abbreviate it.
There you go.
Yeah, there we go. So yeah, about 36 to 50% of the trees were congruent with the fossil order based on that metric. And in this study and later ones, they actually found that using what they think are more realistic metrics, because again, some of these individual metrics have different systematic biases, so you kind of have to treat one separate compared to the other. But overall, these other metrics found that roughly 3/4 of published trees were congruent with the fossil order. So based on all these metrics together, the overall congruence between phylogeny and stratigraphy appears to be very statistically significant. So again, this wouldn't be the case if common ancestry weren't an accurate description of reality, if instead things were independently created. If the flood had happened, how would this have resulted from that? Like,
Right,
This is
because
a
all of the fossils in a flood scenario should be all smushed together
right.
without any significant time ordering.
Or like
But that's
appealing to
obviously
some
not
of
what we
those other
see.
mechanisms, you would expect some like the more buoyant ones to be above the
Right.
denser ones, for example, or all these other patterns. But instead, we see the fossil record is sorted taxonomically based on phylogenetic order that we expect from modern organisms. So why is this the case unless common descent is correct? So
And it's
confirm
worth
prediction.
noting that the evidence, this type of evidence, this concurrence between the fossil record and the phylogenetic um, convergence analysis, coalescence analysis, has actually gotten stronger since the publication of this paper.
Yep.
So, like for example, one of kind of the outliers for a while was the fact that genetically mollusks seem to be kind of this weird divergent group that seems to have diverged earlier than a lot of the other chordate subgroups and like or the bilaterian subgroups. And
Right.
that's
Okay.
weird, right? why did
Right.
why do we see phylogenetically, right? We have, we've got like sponges, we've got cnidarians, like jellyfish, that kind of stuff. Obviously that stuff diverges very early in the history of multicellular animals. And then you have a series of divergence events that actually occur before the so-called Cambrian explosion. I prefer the term Cambrian radiation because that describes
Right.
what the event actually was. It was an example of
Right.
an adaptive radiation. And for a while, it was this weird thing where phylogenetically the mollusks actually didn't line up with the fossil record. We could tell phylogenetically that it sure seemed like they branched off way before we start seeing their fossils. But one thing that's happened over the last 2 decades is that the Cambrian explosion has actually gotten a lot less explodey, right? 5 million years, 10 million years, 20 million years. Now it's a 40 million year event.
Right.
And now it actually lines up better with the phylogenetic evidence than it did 20, 25, 30 years ago. And that's a really cool thing because just, the science of it aside, right? Putting how awesome all the science related to this is. And as we find more fossils from that time period, we're able to expand the range of when different groups diverge from each other. Putting that really cool stuff aside, just keeping this focused on the creationism side of things, creationists love to point to events like the Cambrian explosion, right? And say, look at all this, it doesn't line up with this other thing.
Right.
Well, that's not true, right? Just broadly speaking, for the fossil record, that's not true. There is actually a high degree of correlation between the two independent data sets, but also specifically for events like the Cambrian explosion that creationists love to throw out there, there's actually a high level of correlation, and some of the harder to understand aspects to it have actually, as we found more data in the form of fossils that we didn't know about before, we've actually brought those data sets into closer correlation with each other.
Right. Yeah. And just to kind of add on to that, that's a good point that I also want to touch on was the fact that potentially if we were finding more fossils, so gathering more paleontological data, that one data set, and we were also increasing our understanding of genetics and how molecular sequences evolve, it could have been the case that kind of they were overlapping initially, but then as we got more and more precise in our understanding, they diverged, and it could have been completely off after that. But
Mm-hmm.
instead, as you pointed out, as we found more fossils, as we've understood how to calculate the molecular clock for different organisms, basically, and more robust, as we were mentioning earlier, statistical methods for the more computational power for calculate or inferring these phylogenetic trees. As we've gotten better and better and collected more data, they've still tended to converge with each other. And why would that be the case if separate ancestry was true? Or that this only seems to be consistent even if in fact we're closing in on, so to speak, the actual genealogical history of life on Earth.
We're right. We're kind of shrinking the error bars
Right.
around the correct answer, and it's as you said, this is a prediction, right? And it's becoming more accurate over time as we collect more data, which is the opposite of what you would expect if separate ancestry, especially recent separate ancestry, and a recent worldwide flood was the case, both genetically, phylogenetically, and in terms of the fossil record. What we see is not what we expect, both in absolute terms and in terms of the correlation between those two data sets. Yeah.
Right. And kind of as an aside here, one thing creationists also like to point to with regard to phylogenetics is the supposed places, like we mentioned here, where initially, I mean, even today, there are still some clades, some branches in the tree of life that we can admit are less resolved than others, but there are reasons for that. For example, some speciation events were particularly rapid that can kind of throw off some things. We have ways of adjusting for that, but also we need to accumulate, or we need to gather more data, like more genetic sequences. Like obviously not all organisms out there have had their genome sequenced, and sometimes accounting for certain taxa versus other, poor taxon sampling, for example, those can be important in determining some of these particularly deep phylogenetic relationships, like the deep nodes in the animal tree of life. Yeah, as we've gotten better, a lot of those gaps have been filled. A lot of these polytomies that I mentioned earlier have been more resolved. And again, that isn't the same thing as a complete random jumble of one phylogeny versus another. Like we shouldn't be seeing any agreement whatsoever
Right.
if strict separate ancestry were correct.
And yeah, and you're totally correct about all that. One thing that I do every year for my class is we do a whole unit every spring in my intro bio class, Bio 2. We do a whole unit on biodiversity, and it's in a phylogenetic context. And every year I revise those phylogenies based on the most up-to-date evidence. So I'm always changing the clades that we have in our animal phylogeny. I'm always changing the clades in the eukaryote phylogeny in terms of, whose sister groups to who and which supergroups are present, or no, which ones have been put together, which ones have been broken up. And as we collect more data, we're able to resolve those tensions with a higher degree of certainty and greater resolution. So my hope is that every year, as I update those things, I get closer and closer to what actually happened historically, right?
Right.
And am I ever going to get there in my lifetime? I don't know, maybe. But like every year, I'm a little bit more confident about it compared to the year before, because it's just based on a more complete picture, in terms of just the amount of data we can incorporate into that.
Right. I mean, like as other people have pointed out, a fair point is, there are over 1 million species of metazoans that we've discovered so far, and probably more protistans that we haven't discovered, and bacteria. So there are a lot of organisms out there. So I don't know, like you said, if we're ever going to get the perfect 100% resolved phylogenetic tree of life, but the fact that we seem to be getting closer and closer, getting some of these parts more and more resolved, seems like our methods are working so far.
I mean, yeah, it's and one kind of test of this that's a very famous kind of example of this is, of course, Tiktaalik, right? Because how do we know when that thing should have existed? We can bracket it with other fossils, we can do phylogenetics, and then we can use geology to hypothesize, okay, which rocks are the right age and where should those rocks be located in the world, right? So you have a bunch of different hypotheses that all have to converge in order to make something like that possible, where you have to be able to accurately date the rock formations. You have to be able to accurately evaluate which rocks were coastal, potentially the targets for sedimentation during that time, right, where fossils are likely to form, and not only bracket it with the fossil record, but also phylogenetically, right? We're able to take modern tetrapods, modern fish, and say, okay, how long ago did these things diverge? You put all that stuff together, and we can predict where we should find something that shares a bunch of those traits, a bunch of ancestral traits that are more fish-like, and a bunch of derived traits that are more tetrapod-like. And lo and behold, there was Tiktaalik right where we expected it to be, right? And
Yeah,
these
it's
types
like
of predictions, go ahead.
Or sorry, this like like you said there, it relies on an understanding of geology, biology. So like radiometric dating, for example, if that was unreliable, then why are these separate fields matching up in the first place?
How are we going to know which rocks are which, right? That's how we
Right.
date the formations, right? Creationists often accuse people of, oh, you date the rocks by the fossils and you date the fossils by the
Right.
rocks. No, you date the rocks in an absolute sense. And then once you identify a set of index fossils, then you no longer have to do the radiometric dating because you can use these things that have already been conclusively dated based on the formations they're found in, which
Right.
themselves have an absolute dating radiometrically, right? So like no
Right.
creationist
For
is found
anyone curious,
circular.
there's a whole field about that called biostratigraphy for anyone interested. So yeah, we know how to date these strata. And since I feel like some creationist out there is inevitably going to bring up the fact that we found I there is still some controversy about this. I'll
Yeah,
admit
that's
the Polish
uh-huh.
tetrapod footprints.
Yep.
I was going to bring that. But I just want to mention a crucial point here, which is the fact that even those footprints, assuming they are still early tetrapod footprints, and they do seem to match the morphology of very basal tetrapods that we do have body fossils of. So that's not completely unexpected. But if, for example, we found those footprints 420 million years ago instead of like 390-ish million years ago, before we have any body fossils of sarcopterygian fish in general, like lungfish, that kind of stuff, which actually, as you were mentioning earlier, as we've gotten better molecular data and our techniques with that have improved, I think there is one paper that Erica Gutze-Gibbon brought up in one discussion post, which was they resequenced the data with lungfish specifically and found that the branch point based on that molecular data actually uh, was very congruent with this kind of new revised potential divergence between tetrapods from other tetrapodomorphs in the early to middle Devonian based on these footprints. So now it seems that rather than these two fields diverging as the creationists would like, they're now converging on a new improved answer for the relative sequence of occurrence here.
And it's worth noting that, so I will say I'm agnostic on the Polish footprints.
Yeah.
I just not my field. I don't know. I'm going to wait for the experts to sort that one out. But it is worth noting that if those are in fact kind of early tetrapod traces, they are indicative of morphological traits that are more ancestral and less derived than what we see in Tiktaalik.
Right.
It's not like it's you go back however many million years but jump ahead morphologically. It's that you're environmentally it would be in a similar kind of coastal terrestrial kind of situation.
Right.
But
They're not like reptile trackways.
Right. Morphologically they are less derived than
they're still amphibious organisms, basically. Right. So yeah, that's a good point.
So that is let me scroll back up to the top here. That's Benton and Hitchin congruence between phylogenetic and how do you say that word? Stratigraphic? Stratigraphic data. I don't know fossils. Sorry, everybody. On the history of life. Do we feel like we do? Is there anything else we want to add on that one before we move on to number 3 over here?
Yeah, I was just gonna read the quote, but I feel like since it was on there for a while, people probably
go for it. No, read it. Which I'm assuming it's it's this one. Right?
Just kind of to round this point off. Yeah. Okay, so they say here, even though the results from the SRE test in the present study were not as convincing as earlier analyses, so that's what I mentioned earlier about the slightly lower percentage of congruent trees. Quote, the RCI and SCI metrics showed impressive left skewing. The majority of cladograms tested showed good congruence between cladistic and stratigraphic information. Cladists and stratigraphers may breathe easy. The cladistic method appears, on the whole, to be finding phylogenies that may be close to the true phylogeny of life, and the sequence of fossils in the rocks is not misleading. So there you go.
I love it.
Okay.
And as we talked about, that has only gotten more true since this paper
Right.
was
Exactly.
published, which is great. All right, so let me take that one down. Now we're going to zoom in, and we are going to talk about some molecular stuff because now we're going to
Talk
do
about fossils, now
we're
molecular data.
we're doing cytochrome C.
Yes, cytochrome C.
Cytochrome C. Let me pull that one up here. All
And
right,
I
so let
will
me.
note that what I'm about to show about cytochrome C protein is not unique to it. These have been done for other proteins as well, but I'll touch on that in a moment.
This is an early example, which is very
Right,
cool.
right. It's like we knew about this since I think this one was from 1963. So we've known about these relationships
All
for a
right,
while.
we are doing this one. Here we go. All right, so this paper, primary structure and evolution of cytochrome C. What are we looking at here?
Okay, so if you want to scroll down, I'll say stop when you get there.
You tell me when to stop.
I'm gonna keep going. There's one specific table I want to highlight.
This
All right,
is
there we
the
go.
table you want right
Yep.
here. Yep. That's the one.
Yeah, beauty right there. Okay, so the argument here is a bit of both induction and deduction. So and actually, I will note that a lot of these arguments and points I'm presenting have already been discussed on the Talk Origins website. That's a for anyone who hasn't looked at those, that's a really good source of information for all these creationist claims, lines of evidence for evolution. So I encourage folks to go check that out.
I'll just say real quick on Talk Origins, it is terribly outdated. But the hilarious thing is that it still works for like 90% of creationist
Right.
arguments because they've been saying the
Right.
same thing since at least like 2005. There are
They haven't
very
updated any of
few.
their arguments.
There are very, very few creationist arguments that are newer than 2005.
Right.
Very, very few. Even I'll give you a really funny example that I was just like, I don't have to do anything about this. Discovery Institute, Intelligent Design, C Design Proponentist think tank, just I think today, it was either today or yesterday, put out on YouTube a video about how the bacterial flagellum couldn't evolve. And I'm like, we've done this. You can just stop doing that
That's pretty
anytime
funny.
you want. So while Talk Origins is not going to have, like, I don't think, for example, I don't think created heterozygosity is in Talk Origins.
Yeah, I don't think so.
I don't think so. But like most other stuff is, you have to get quite recent, a little bit into the 21st century to not be found.
Right. Look, they have a few articles on there about dinosaur soft tissue, so
it's right. So they've caught some of that.
So,
Yep.
and that's one of the more recent creationist arguments.
Yeah.
So
this paper is from 1963, but this is an early example of something that we've now done a lot. So with that out of the way, what are we talking about? What is cytochrome C? What the heck are we doing here?
Yeah, so cytochrome C is basically a protein found in many different organisms, eukaryotes, prokaryotes, so bacteria, animals, fungi like yeast, plants, all these different organisms need it because it has a basic function in cellular respiration, the pathways involved with that. It's, I think, specifically involved with the electron transport chain.
Mm-hmm. Yeah, it's in the mitochondrial inner membrane.
Right.
It's an ETC component. Yep.
Right. Yeah, so it has a very basic cellular function. So if you don't have this protein, or you have a radically different version of it, then it's not going to work. The organism's going to die, right? So that's one point in the argument here. So there are kind of a few premises that make this argument pretty strong, I think. And this is one of the first ones, that cytochrome C and these other what are called ubiquitous proteins, that's the term Talk Origins use, in their article, are basically found in all these different organisms, and they perform these very basic, necessary to be alive kind of functions. So clearly, if you're a human, or if you're a chimp, or you're a fish, then whether you have a unique version of this protein or not, like the protein itself is there irrespective of the specific morphology or environment that particular organism is living in. So it's species independent, basically. It doesn't
Yeah,
depend
it
on what
does.
kind of organism you are. You have
It
these proteins.
Yeah, it does a very specific, very important job, and it does that same job whether you're Homo sapiens or
Right,
some kind of fish or jellyfish or E. coli. Like cytochrome C oxidase does the same thing.
right. And same with the DNA replication mechanisms, etc. So that's one point in the argument here. The second point is that proteins in general are rather flexible, molecularly speaking. You can switch a bunch of the so just a quick primer here. So the DNA codes for messenger RNA, which gets translated into a specific amino acid sequence, and that amino acid sequence folds on itself into a protein. And specifically, the part of the protein that kind of, well, for most enzymes at least, that has this enzymatic activity is called the active site. So that active site in the protein, when it eventually folds up on itself, the amino acids there have to be a certain order and sequence of amino acids. Otherwise, the protein overall isn't that particular protein isn't going to be able to perform that function that the cell needs. So if you have a change in an amino acid there in that active site to a chemically different amino acid, say from a hydrophilic, water-loving amino acid to a hydrophobic, water-hating one, that's going to mess up the protein. It's not going to be able to do its function. The organism dies, basically. So that part of the protein is under purifying selection. Whereas other parts in that protein, the ones that aren't involved in that enzymatic activity in the active site, those are basically functionally silent. It doesn't matter if you have this particular amino acid there or this particular amino acid or a different one. That doesn't really affect the overall protein function. There are some parts of the amino acid sequence that you have to have a certain length of amino acids there, otherwise it's not going to then fold properly on itself and won't be able to function. So that's one structural constraint there. But generally speaking, most of the amino acids in most proteins can change quite a bit without affecting the overall protein structure. So those are silent mutations, basically.
And there's even some mutations where, or even some sites within a polypeptide where the specific amino acid doesn't particularly matter, but like as long as it's small and non-polar, it works
Right.
fine. And any one of a half dozen amino acids will work in that position, or it's one of the bigger ones. As long as it kind of fits that spot, it'll work, but it doesn't have to be this specific one. It can be one
Right.
of a set, right? So you have relaxed selection over a lot of that structure.
Right. So yeah, if anyone wants to look up an amino acid chart, like Dr. Dan said, you have a bunch of hydrophilic ones, hydrophobic ones, negatively charged ones. There are a bunch of different amino acids of the 20 overall within each category. So that's the second point of the argument, is that a lot of the structure of cytochrome C here, for example, is those particular amino acids there are not functionally essential for the protein to do its job. Only,
Mm-hmm.
I think, is like about a third of cytochrome C, the amino acid sequence there, is functionally necessary for it to have this function in the electron transport chain. And in fact, another example here, just to kind of reinforce this point, is that on Talk Origins, they mentioned human hemoglobin.
Mm-hmm. Perfect
Roughly three,
example. Yep.
right, roughly 3% of the world's population. There are at least 250 or more different amino acid mutations that you can have that are carried by different people. So there's a lot of flexibility in hemoglobin and these other proteins, and they don't seem to have a lot of uh, negative effects on your medical health or the health of the organism that we're talking about. So yeah, it's very well tested concept that a lot of the amino acids in these proteins are functionally silent, basically.
So then let's bring this to this table, because this table is beautiful. Also, you'll
Yeah.
note that, man, was it 1963 when they wrote this? Because we're not using we're not being very specific here. We're using horse and man for comparison, which is
Right. Yeah.
not great. But what we can see here, just to kind of walk people through this a little bit, is what you have is, so horses and humans, right, are mammals. And we know when they diverge from each other. Now, this is from 1963. This is based purely on the fossil record. So some of these numbers are not quite, like this one's going to be an underestimate relative to what we know now. But then you've got horses and pigs, which are much more closely related to each other than either of them are to humans. So that's a much more recent divergence. Then you have horses, pigs, rabbits, and humans to chickens. Now, a chicken is a bird, so all of those mammals are equally related to birds in terms of the time since that common ancestor diverged, right? They all share the same most recent common ancestor.
The technical term is equidistant, I believe, right?
Yes. So we can say, so this estimate, 108 to 150 million years, that applies to all four of those comparisons. And then we take those five things. I loved how they do this. It's nested hierarchically, right? And we take these five, which are all vertebrates, and we can, which are all tetrapods, and we compare them to tuna, right, which is obviously a marine fish. So they all share the same most recent common ancestor. And then you take all of those vertebrates and you compare them to yeast, which is a fungus. Now, this number is too small, right, because we know subsequently that the fungal lineage diverged from what became the animal lineage more than 520 million years ago, because that puts you smack in the middle of the Cambrian. But at the time, that was the estimate they had. But the important thing here are these numbers. Because what happens if we were to plot these numbers against these numbers?
Amount of difference correlates with the amount of divergence.
There you go. You get a nice, neat correlation between all of those things. And we're not looking at genomics, right? We're not looking at genome sequences. We're looking at the amino acid variants in the protein, which, as Andrew explained, you actually have a lot more flexibility in that. So you might have something where genomically something might be very similar, right? 70 or 80% similarity. But if you look at the actual amino acid sequence in this or that protein for those two organisms, you might find that it's 20%, 30%, maybe even less, depending on the context, right? Because there's only a very small part that has to be the same in order for that protein to function. Sometimes it's more constrained than others, right? So like there are certain types of proteins that are more like structural, where I'm thinking, histones, or I'm thinking about connective tissue, like fiber kind of things, like collagen fibers, which is a triplex of it's a triple helix, where those things need to be able to associate with each other up and down the length of that. So that's a little more tightly constrained in some ways, whereas something like an enzyme, or as you said, hemoglobin's a great example of this. There are a few small parts of it that really need to work a certain way, but the rest of it, as long as the shape and the external properties are correct, then you're pretty much okay. And this table is showing then kind of approximately neutral accumulation of amino acid changes in these proteins, correlating that with time to most recent common ancestor in the same way that we can do it with genomics, with DNA sequences.
Right. Forms a nested hierarchy.
Bingo. And
this, I guess, just for audience sake, should we briefly refresh on what nested hierarchies are?
Yes, let's do that. Would you like to do that, or should I do that?
I feel like you could probably articulate it better.
I'm happy to do it, although I'm sure you're able to do it too, because I've talked to you about this a lot, and I know you can explain it. So a nested hierarchy is, I always describe this in class as a box in a box system. In fact, if we're it's a little looser tonight, so give me 1 second. I should be able to pull up a figure of that I use in class to illustrate this a little bit. Just what do we mean by a nested hierarchy? So just give me a second here to find the the correct figure. Here we go. We'll use this one. This will work fine.
While you're
This is a
bringing that
yeah. Go
up,
ahead.
one number I did actually want to quickly mention based on what you just said about, or just kind of reinforce this point about how incredibly flexible some of these proteins are. Like you said, it does vary between proteins, but in general, I found this particular calculation based on relatively modern mutation analyses, kind of changing different parts of the protein. They calculated that there are at least roughly 10 to the 93rd power different possible functional cytochrome C protein sequences that would be roughly functionally equivalent, or maybe a little more efficient, maybe a little less efficient, but for all intents and purposes, the same. So the point is, like, if separate ancestry were true, they should be random with respect to each other, because these organisms don't share a common lineage. They didn't inherit the same mutations. They don't follow the hierarchy.
Bingo. All the existing variation would have to have either one, been put in place, which is an untestable hypothesis, or two, accumulated since they were created, right? And they should be though any mutations that occurred since the creation event, the separate creation, should be uncorrelated with each other. We shouldn't get a nested hierarchy. Exactly. And that works for DNA, and it works for amino acids. Yep. And so let me just pull up this figure real fast. I'm gonna take this down, and I'm going to switch to something else very quickly, and then we'll switch back just to show what we mean by a nested hierarchy. Is this is a slide I actually use. It would help if I added it to the stream. This is a slide I actually use in class when I introduce the concept of phylogenetics. And it's the idea that we want the way we name things and describe things to reflect their evolutionary history in such a way that as you go from a small group, like a genus, up to a larger group, family, order, class, right? As you go bigger and bigger, you're including more organisms in that group, and you're including more distant common ancestors in that group. So in this figure right here, right, each purple box represents a species. Each green box represents a single genus. Red is family, and then the blue box is a single order. And you can see that, like, this genus this genus right here, right, that has a most recent common ancestor, this node right here, compared to the most recent common ancestor for this family on the right. So this is what we refer to as a nested hierarchy. It's a box in a box system where you start small, and as you go up and up and up, you're grouped in increasingly large groups that encompass the smaller groups in the next level down and all of their included groups. Right. So this is the idea of monophily. Right. It's a hierarchically arranged system. Okay. So that's what we mean when we're talking about nested hierarchy.
To the cytochrome C thing, if you can just leave this figure.
Oh, sorry. I'll put it back. There you go.
To use it for illustrative purposes. So yeah, if you look at this figure, going back to the cytochrome C thing, if, say, all the three genera depicted here were independently created and have separate ancestry with respect to one another, there is no reason that the two genera on the right-hand side of the screen there would have fewer amino acid differences between them compared to the third one. So there's no reason that they should appear as though one branch multiplied into another in terms of these amino acid differences.
Exactly. Once you get outside the created groups, they should just be all jumbled up. They should just be uncorrelated with each other. You shouldn't be able to continue generating that nested hierarchical system, which, as we talked about at the beginning tonight with Hillis et al. 1992, is actually a reflection of the evolutionary history. Right. So the presence of that nested hierarchical pattern in groups beyond what young Earth creationists would say are the separately created kinds, right? The presence of that organizational system, that nested hierarchy beyond those groups, or I guess a better way to say it would be across those groups when you encompass two of them or more, that's evidence against separate creation, right? Because you shouldn't be able to resolve those separate groups when you cross the kind boundaries.
Right. We would say the only way to correlate these essentially silent changes is shared inheritance, aka common ancestry.
Exactly.
Actually, something this is actually segues into the main creationist argument I've heard, especially when I brought this up with SFT. If you don't mind highlighting Nestlig's second to most recent
Oh,
comment
yeah, hang
here.
on here. Here we go. This one.
Yeah, so that's a good point there, Nestlig, because like I said, when I brought this particular argument of cytochrome C up with SFT in a late night discussion, it seemed to me the only major counter he had to it was related to, I think, something Nathaniel Jeanson brought up in one of his books, which is this idea of what's called protein moonlighting. And
Mm-hmm.
he hypothesized, loosely there, that the reason these differences exist is because the changes there are not actually silent. Instead, they actually reflect a different secondary function that does differ between these broad groups of organisms. I'll give my thoughts on the problems with that, and then if you want to, Dan, you can
Yeah,
give
go
your
for
thoughts.
it. Go for it.
So first, that kind of ignores the first premise we talked about with this argument earlier, which is these or the same protein is found in many different groups of organisms. So it seems like it would be a heck of a coincidence that they would just happen to fit this deep nested hierarchy of these different changes if like it would have to be fine-tuned in such a way between humans and chimps, for example. Like, do they really live in similar environments that they would need to have zero amino acid differences in this particular protein that doesn't really affect their overall morphology? Like, it would just have to be it, as others have pointed out, it would have to be almost as if the designer was trying to deceive us into believing evolution.
Mm-hmm. And my counter argument to that silly argument from Jeanson is that, as Jeanson should know, he may not, but he should know that we can actually measure the degree of selective constraint on an amino acid sequence, because we can calculate this thing called the DN-DS ratio. It's the rate of
Yes.
non-synonymous changes compared to the rate of synonymous changes that occur in a region of the genome. And if you have a region where that ratio is approximately 1, then that is indicative of approximately neutral accumulation of changes in that region. Whereas if that number is below 1, that means we're suppressing non-synonymous changes, meaning mutations that change the amino acid sequence. And if we see selection against that, that's purifying selection. That means that that region has to be the way it is. And like we've been talking about, you can look across these proteins and you actually find, according to the DNDS ratio, you actually find that there's a lot of approximately neutral evolution happening, right? Which means
Right.
the specific amino acids are not tightly constrained, which, if Jeanson is correct, you would need the specific amino acids to be tightly constrained, because these sites that differ between organisms that are closely related, they would be different because they're doing some specific thing that requires them to be different. Well, that's
Right.
incompatible with the DNDS ratio of approximately 1.
Right. And actually, one other thing I have to counter that silly argument is the fact that we've actually indirectly kind of tested this, because we've taken yeast cells and basically taken their native cytochrome C gene, removed it from them, and then inserted the cytochrome C gene from, let's see, humans, horses, rats, tuna, fruit flies, and pigeons into those yeast. And what was the outcome? The yeast survived.
Yeah, it works.
These differences are inconsequential to the survival of these individual organisms. Again, because they only the protein is only seems to only be performing this essential cellular function.
Yeah,
It's
it's
not
not in the
influencing
regions.
anything else.
Right. It's not, those mutations are accumulating in the regions that are not constrained, because they're not the regions that are required to do the electron transport function, and they're
Right.
also not doing some other important thing. Exactly.
Right.
Yes.
Yep.
So let's talk about. We're going to finish
Drum
up. We have
roll.
one more topic, everybody. And we are going to talk about pseudogenes, and I'm really excited about this.
Good
So
old
let me
pseudogene.
share this one. It's this one right here.
Essentially an oldie but a goodie, but with a little twist on it.
Let's do it. Let's talk about pseudogenes.
So there are actually many different types of pseudogenes. So there are pseudogenes that, for example, vitamin C related to humans and primates. You have the gene, it accumulates mutations, but it's not affecting the overall fitness of the organism. So it just accumulates mutations. You have a single copy of a broken pseudogene, basically. In other cases, you can have duplications of one gene, the original copy that was copied. Retains the original function. It's there, but these other copies that were duplicated from it, they're redundant. Basically, they can accumulate their own mutations, which, as we've seen with other organisms, also happen to follow a phylogeny. So there's another line of evidence there. And finally, there are what are called process pseudogenes. So in a very rare occasion, you have a gene that, of course, is transcribed into messenger RNA. And during that transcription process, the original promoter, basically, in all the kind of regions upstream and downstream of the gene, they're not incorporated into the messenger RNA. And of course, the introns are spliced out later. So you just have basically the gene, the RNA version of the gene there. And in a very long while, you can have that mRNA be reverse transcribed back into DNA and inserted in essentially a random part of the genome. So these are what are called process pseudogenes, and sometimes even those process pseudogenes can then be co-opted into what are called retrogenes. But again, that's another topic. That's another source of evolutionary novelty there.
I love biology. It's so weird. It's wonderful. I just love that weird stuff that can happen. Anyway, continue. That's a side story. Continue. So we
Yeah,
get processed pseudogenes.
right. So one particular example of shared process pseudogenes that I like are actually this family of pseudogenes called nanog, and I think it's the name comes from a place. I think it's a Nordic place. The guy who discovered the original gene, the functional gene, was he found this gene, saw it was related to stem cell health, I believe, and came up with this name nanog based on a Nordic text which mentioned this place of kind of eternal life kind of thing. Anyway, that's a side story. Just interesting history there. But basically, you have the nanog gene, the functional version of the gene, as in all these different primates, and still does this function. But in humans, chimps, and at least the other great apes that we have genome sequenced, there are 10 other pseudogenes, process pseudogenes of the nanog gene that seem to be inserted in random places in the genome. And when we compare the pseudogenes, we can actually see kind of a similar story to ERVs that have been talked about in other videos, that they're in the same chromosome locations between humans, chimps, and all these other great ape species because they were inherited from our most recent common ancestor in those locations where they are already inserted. So shared insertion sites. And then, of course, on top of that, similar to ERVs, we have shared mutation. Yeah, there we go. There we go. That's the good graphic there. Yeah, so on top of that, those shared chromosome locations that they were inserted in, they also have a shared mutational history, which we can use to document, okay, before the split between all of the modern great apes, what was the first pseudogene inserted, what was the next one, what was the next one. And in this paper, they've actually done just that and found out the mutational history of when these different pseudogenes were inherited or inserted and then inherited, basically.
Which is fantastic because it gives us not just a kind of a single point of here it is in this one, here it is in that one. And maybe it's in gorillas or maybe not, or maybe it's in humans, chimps, gorillas, but not orangutans, right? We can use these, as you've alluded to earlier, we can use these things, these kind of remnants of other coding sequences, whether it's pseudogenes, processed pseudogenes, endogenous retroviruses, right? We can use these things to actually reconstruct the story. And the beautiful thing with a situation like this, where you have so many of them that clearly share the same evolutionary history, you're not just looking at one point, you're looking at an accumulation of points over the course of the shared evolutionary history of two or more species of great ape, which is really cool that we can do
it
this.
just so happens to match the phylogeny of apes inferred from morphology, from regular gene sequences. It all just perfectly matches up. So we are in fact apes. That's the message here. We're very confident of that conclusion.
And I just want to point out just a small little Easter egg in this table for everybody right here. If you look right here, right there, we've got nanog P2, right? You've got chromosome 2 in the human, chromosome 2B in the chimp, because remember, you've got that fusion event. And that's another piece of evidence, right, for the fusion, right? It's not just that you can identify the centromere, you can identify the telomeres, it's that you can actually see how the actual content of those chromosomes lines up side by side. Just a neat little tidbit buried in that table for everyone to see.
Right. So for these processed pseudogenes, not just nanog, but in general, but speaking about nanog specifically here, there are a few details that I want to reinforce here that make what I think this argument particularly strong compared to, say, creationists who will say some pseudogenes are functional, or they may have functions or be transcribed or regulate other things, etc., etc. One is the fact that, as I talked about previously, when these processed pseudogenes are inherited, they're reverse transcribed from an mRNA product that was already what's the technical
it already gone through spliced.
Right. So yeah. So it doesn't have a promoter.
It's processed.
Processed. Yeah. Yeah. So
it's transcribed and then spliced. So you're missing a bunch of stuff that you would need. So then it re-enters the genome without all the stuff.
Right. So without a promoter also entering in with it, it can only be transcribed spuriously, I would imagine. So unless it can kind of show that somehow it's essential spurious transcription, or that it's being transcribed not spuriously somehow, I think that's a pretty good indicator that these pseudogenes are in fact essentially broken and therefore are functionally neutral, basically. So I think that's a good part of the argument. But what do you think?
Dr. I 100% agree that talking about junk DNA, I just talked about this with Dr. Casey Luskin from Discovery Institute last week as we sit here and record this. And one of the things with so many pseudogenes that you find is they're actually not transcription competent. You could find the sequence there, but like the promoter at some point has gotten deleted or mutated, so it doesn't work as a promoter anymore. Or in the case of processed pseudogenes, it's just the coding sequence that lands somewhere else in the genome, unassociated. There's no associated promoter at all in many of them. Well, how's that thing supposed to function? All the proposed functions for pseudogenes are that they operate as RNA, and the RNA of the pseudogene then does something. Well, like if the pseudogene doesn't get transcribed because there's no promoter associated with it, what are we doing? It's not going to be functional, which means when we're looking at this nested hierarchical pattern, we're looking at things that are just happening in the genome stochastically, right? The reinsertions or the inactivation of the pseudogenes. And those things are going to accumulate in approximately a neutral fashion, which means we can use them very effectively to reconstruct our hierarchies. And the mechanisms involved here, and this is something that I think we've kind of hit on a couple times tonight, is that the mechanisms involved here are independent of the other things that we can use to infer universal common ancestry, right? We
Right.
can use the pseudogenes and specifically the processed pseudogenes. That's a different thing from using endogenous retroviruses. Those are independent mechanisms that occur in the genome, right? They're
Right.
uncorrelated with each other.
Or
You can look
fossils.
right. So you have these different lines of evidence. It's the genomic equivalent of using multiple different types of radiometric dating to arrive at the same date, is what we're doing. We're able to do that genomically using all these different kind of aspects to genome sequences and all these different types of elements.
Right. And as Nestle also points out here, I think another good point, of course, is the reverse transcription and integration process is not locus specific, just like with ERVs. So that's,
Exactly.
uh, in
If you
case anyone
share
doubts that, then yeah.
Yep. There are certainly hot spots and cold spots in the genome. Like it's
Right.
hard to insert something in a region that is persistently heterochromatin, for example, right? Where the DNA is all coiled up. It's hard to insert something into that. So you have hot spots and cold spots, but the specific site of insertion is stochastic. It's probabilistic. It's not like I'm going to put it right there and I'm going to do it every time. So if you have a specific pseudogene and specifically a specific processed pseudogene in the same place across multiple genomes, that's going to be very strong evidence of the common ancestry of those genomes and the occurrence of that insertion in the common ancestor.
Especially when all the purported functions are mostly for, like you said, or uh, pseudogenes that could potentially regulate or endogenous retroviruses that can be transcribed. But so far, as far as I'm aware, no researchers have found these same purported functions for processed pseudogenes and the like. So,
Not to my knowledge, they haven't.
yeah.
And that's not to say that they have. I just, not to my knowledge, is what I'll say.
Right.
And I was researching this quite a bit over the last couple of weeks in preparation for my chat with Dr. Luskin. So I don't know of any examples. That doesn't mean there aren't any, but I am not aware of any in
Right.
the literature.
As far as we can tell, yeah. And I think that's actually a good point to, or actually one thing, and then I think that's a good point to wrap things up on, is just quickly mentioning there's actually a functional version besides the original version of the nanog. However, it's only found in humans and I believe Neanderthal genomes. So there's the version of that there. It is functional and it
Mhm.
kind of recognizable to these other pseudogenes, but seemingly these other pseudogenes don't have any function. So why is that? I don't know.
Yeah.
That's kind of
So,
a conundrum there.
and you mentioned that earlier, where oftentimes a gene will duplicate, and then you'll have two potentially functional copies, right? And then you're only selectively constraining one of them. So whichever
Right.
one inactivates via mutation first, you're then going to have purifying selection maintaining the other one. And then the broken one now is just going to mutate more or less freely because you don't need it.
Right.
So what it talking about having all of these different, examples of this, we can look at these insertions and we can point to both a retro insertion, right, a process pseudogene that appeared in the human genome since our split with chimps. And you can also look at a functional duplication rather than a process pseudogene, but that region, including the promoter, that occurred in our common ancestor, at the very least, going back with Neanderthals.
Right.
So that's going back probably over couple million years for wherever. I think people who know better than me can correct me on this, but I think that would have been probably Heidelbergensis. So figure out when
Yeah,
that
at
would
least
be.
a few 100,000 years, I would reckon.
Oh yeah, definitely a few 100,000 because it would have to be
Yeah.
before the out of Africa event that led to Neanderthals, right?
Right.
So yeah, and that's it's being able to reconstruct that exact history is very cool. And this pseudogene, this gene and pseudogene family is a really great example of that.
Right. Yeah, and I just point out, I think the specific one that I'm talking about there is the Nanog P8 version there. And like I said, it's only present in humans. I just point that out because inevitably some creationist is going to suppose that somehow these pseudogenes do have a function. Well, okay, why do they look the same and yet some have this function but not this other function or no function at all? It kind of doesn't really make coherent sense.
But it makes perfect sense in the context of common ancestry of
Right.
humans and all of the other apes. Then it
Right.
makes sense, right? That's exactly what we expect to see. With separate ancestry of humans and the apes, none of this makes any sense.
Right. And I think that's a really big point to take away from this discussion here tonight is all these lines of evidence could have disagreed with one another. The sign that calm descent is a good working theory and it makes accurate predictions is they all come up with the same or essentially the same nested hierarchy, the same pattern of relationships. And yeah, I think that, like we said with the cytochrome C and this, for example, the only counter that I've heard is this appeal to unknown or untested functions, like junk DNA in general, that they haven't yet put in the evidence to support their claims yet. So why should we kind of? I mean, we are still testing it, obviously, with each genomic segment, but if they the burden of proof is on them basically. So they need to go out and test it. They need to show these purported functions. Otherwise, why should I believe you?
Yeah. Every one of these things, like that paper we just talked about, every one of those is a test of an evolutionary prediction. And what we keep finding every time is that the data are consistent with the evolutionary prediction.
Right.
So I think that's a great place to end it. Oh yeah, sorry. Here, that functional
right,
one, is it RNA or protein coding? Do you know? Because I don't know. The one that we share with Neanderthals.
The nanog P8. I don't know actually. I'll have to look into that.
P8, is that going to be processed? I don't know this exactly. I don't know this. I wasn't familiar with this paper until you sent it to me.
I believe it's a retrogene, so it was processed and then co-opted, as far as I know.
Oh, so it's probably a functional. So it makes sense to be as an RNA then. It would be a long non-coding RNA. I believe is what I would expect in that case. Okay, cool. There's your answer, people. We think, yes, long non-coding RNA is what we think. But again, I didn't know of this paper until a couple days ago. So I can.
Well, I believe you're going to be posting some of these papers. So if anyone wants to go
have the links in the description shortly,
in case we
once we finish this up. All right, so thank you, Andrew. I learned a ton from that. Thank you for bringing some of these papers. I love the historical examples that illustrate this stuff. And it also serves to just point out that there was never a time when any of these creationist arguments made sense. It just wasn't a thing ever. So thank you for coming on. You are a frequent, just quick correction from Nestle. Paper says it's a protein product. There you go. Okay. We'll go with that.
I'm a mistake. Sorry.
See, we're all learning together. This is what we do here. This is great. So Andrew, you are a frequent guest and contributor all over the evolution and creationist sphere on YouTube. So do you have anything specific coming up, or should people just be on the lookout for you whenever, you can't resist the siren call of SFT saying something dumb?
Yeah, I guess there's I know SFT's doing a few geology streams in the future with caves and how they formed during a flood. That should be interesting from one of my interests in geology, especially besides evolution. I don't everyone can probably catch me in some future streams over on Answers in Atheism. We do some streams over there trying to get back into the scientific as well as philosophy side of things. So I think that's about it. Yep.
Okay, great. And for this channel, so next Wednesday, that is the 15th of May, 2024, I am having Sal Cordova on, and we are going to talk about his protein orchards argument. So the kind of argument that he makes is that proteins do not all share common ancestry. There's not a single common ancestor protein in the same way that there is a single common ancestor cellular population, right? And Sal looks at these protein families that are separate from each other and takes that as evidence against common ancestry and specifically leans on the probabilities of these different protein families. And so that YouTube stream is actually available right now. There's Sal's kind of verbiage that he gave me is in the description of the video. So that'll be next Wednesday. And then the week after that, I have a pre-recorded video that it is, for right now, it is future recorded, but it will be pre-recorded that I believe is going to be a creationist contradiction unless something else comes up in the meantime that I want to slot in there. And then after that, I've got a couple of options. Might do a chapter out of the New Answers book, some Answers of Genesis. I've got one on carbon dating next, and so I may do that. So that's what I have coming up. We're going to chat with Sal Cordova is the main thing, followed by some of my more usual stuff, contradictions and AIG saying wrong things. So that's where we're going to leave it, everybody. Andrew, thank you so
Awesome.
much again for doing this and doing all the legwork, putting together these papers for us. I'm going to edit the description to include the papers. So they're going to be linked down below if you want to dig into any of the stuff we talked about tonight. And with that, we are going to call it. Thank you all again, everybody. Have a great night.
Keep learning.